Okay, let's dive deep. We're tackling a stack of sources today that zoom in on, well, a truly microscopic challenge, literally. We're looking at how AI is being pushed, really pushed, to reconstruct these incredibly complex biological structures, all from images taken with electron microscopes.
That's right. Think of it like trying to build a super precise 3D model of, say, tangled spaghetti. But the spaghetti is basically invisible except when you look at these ultra-detailed black and white slices. So the mission here is really to unpack these source materials. They essentially lay out a specific research problem in neuroscience and a plan to solve it.
And the core subject.
It's all about using AI for the detailed 3D reconstruction of Schwann cells. They're a specific type of nerve cell.
Okay, and the big goal, the one that comes through in these sources, it seems pretty ambitious.
It is. They want to build their own system, an open source in-house AI, basically to replace the expensive commercial services they're using right now for analyzing these images.
Gotcha. So let's talk about the research problem itself. What are the sources saying is the fundamental challenge?
It really boils down to taking these flat 2D images, continuous slices from an electron microscope, and getting a computer program to figure out the complex, interwoven 3D shape of these nerve cells.
And the technical puzzle is automating that.
Yeah.
Identifying and tracking the cell boundaries.
Exactly. Those faint lines in the black and white images, you need to track them across maybe thousands of slices.
Right. It's one thing to see it in one picture, but following a single cell as it twists and turns through that whole stack. Wow.
Mm-hmm. And they're not just thinking about one cell reconstruction either. The scale they're aiming for, potentially, is reconstructing all 60 or 70 nerve units in a complete organ sample.
All 60 or 70.
Yeah, imagine the sheer amount of detailed structure you need to capture there.
Which brings us to the difference between how they do things now versus how they want to.
Definitely. Right now, even using a commercial AI service, they mentioned one called WebNosos, it still needs a lot of manual correction afterwards.
Oh, wait.
What they're aiming for is something much closer to a fully automated, autonomous AI system.
Mm-hmm. And you can see why when you look at the time involved.
Oh, absolutely. The sources mention reconstructing a single cell structure manually. That can take literally hundreds of hours.
Hundreds.
Hundreds. And even with the current AI help, they're still putting in, 10 to 20 hours of manual correction per cell?
So the potential time savings are just enormous if they can automate this.
Staggering, yeah.
Okay, so what's the raw material? What does this data actually look like? Electron microscope images, you said?
Yep, electron microscope continuous slice images.
And these aren't like regular photos?
Not at all. They're black and white, yes, but the resolution is kind of mind-boggling. We're talking a 4.2 nanometer scale.
4.2 nanometers. That's tiny.
It's incredibly small. Like, if human hair is about 80,000 nanometers wide, so you're seeing detail almost down at the molecular level. And the biological structure itself is just incredibly complex, super finely detailed.
Plus the sheer amount of data.
Oh, yeah. A single sample, they say, is maybe 2,000 to 3,000 individual images.
2,000 to 3,000 pictures for one sample.
Right. And they expect the full project might involve data sets 10 times larger than that.
So tens of thousands of these super high-res images.
Yeah.
All needing to be processed in sequence.
Exactly. And visually, the cells themselves are a huge challenge. They have these highly irregular, incredibly tangled shapes. And while the images are sequential, so they change gradually slice to slice.
Capturing those subtle changes automatically, that's the hard part.
That's precisely where the difficulty lies, yeah.
Okay, so you've got these incredibly detailed black and white pictures, super tangled structures, thousands of slices, which leads us to the core question. Why is this specific problem so hard for AI?
Well, those data characteristics we just talked about, they're really the heart of the technical puzzle. I mean, first, these nerve cell structures are so irregular, you can't just tell the AI, look for circles or look for squares. The shapes are way too complex, unique.
And there's that tricky part you mentioned earlier, the touching but not overlapping.
Yes. That makes it so much harder than just segmenting, different rocks in a picture. These cells touch. They intertwine constantly like the threads in a tight ball of yarn, but biologically they don't actually merge or overlap.
Oh.
Distinguishing the boundary where two cells are just pressed right up against each other versus, say, a fold within a single cell's membrane. That's a really subtle visual difference. Super hard for an AI to just learn automatically.
Okay, that makes sense.
Where?
What else makes it difficult?
Well, beyond the shapes, there's just the sheer computational load.
The processing power needed.
Exactly. Analyzing millions upon millions of pixels across thousands of images. Finding these tiny structural details and then tracking them in 3D. That takes enormous processing power.
And then there's another layer of complexity the sources mentioned,
Yeah.
the lack of standardized criteria.
There aren't always truly standardized objective rules for defining the precise morphology, the shape of these specific cells. So what one expert biologist might define as part of one structure, another might interpret slightly differently.
Which makes training an AI much harder, right? If the correct examples it learns from are perfectly consistent.
Precisely. How does the AI know which version is right if the experts themselves might vary slightly?
So an AI model that can tackle this needs some special skills.
It really does. Obviously, it has to handle those crazy irregular shapes we talked about. But critically, it needs to track the boundaries and predict how the structure sort of deforms and shifts from one 2D slice to the next. That's how you build the accurate 3D model.
And because of all that biological complexity and the lack of perfect rules, you absolutely need the human experts involved.
Absolutely critical. You need to integrate expert biological knowledge, not just for the initial training, but crucially for validation all the way through the process. You need that sanity check.
Okay, so these are some pretty significant hurdles, but the sources do offer a way forward, right? They assess the feasibility.
They do. And the good news, or the conclusion they reach, is that automatic segmentation from these black and white images is considered technically possible.
Ah, okay, so the mountain looks climbable, maybe?
They seem to think so. And they have a starting point. They mention using existing labeled data. Apparently, they've already got 7 cells painstakingly segmented across hundreds of images.
7 cells? That doesn't sound like a lot for deep learning.
It's not massive, no, and they acknowledge they'll definitely need more. But it's a crucial base to start from. It proves the concept.
And how do they suggest the AI learns from this?
Well, they suggest leveraging common computer vision techniques. Things like analyzing image contrast, using feature learning.
Feature learning, like training the AI to recognize the visual patterns that define those tricky cell boundaries.
Exactly. Rather than trying to program it with explicit geometric rules, which wouldn't work for these shapes anyway, let the AI learn the patterns itself.
Okay. And their strategy involves breaking this whole big problem down.
Into phases, a phased approach.
Phase 1.
Phase 1 is building the basic segmentation system. This is where they take that existing labeled data, apply deep learning, teach the AI to recognize features and boundaries in those 2D black and white images. The goal here is to automate that initial labeling process for each slice.
Makes sense. Start flat, then?
Then phase 2 is continuous tracking. This part uses the fact that adjacent image slices are very similar. The AI needs to predict how the cell sort of deforms or changes shape just slightly from one slice to the very next one.
How does it check if it's right?
They suggest maybe using physical or biological constraints. Like,
Yeah.
a cell isn't just going to spontaneously disappear or teleport across the image between slices. Those kinds of real-world rules can help validate that the tracking is plausible.
Okay. Segmentation, then tracking was phase 3.
Phase 3 is quality control, and this is absolutely vital. They need to figure out objective ways, maybe using machine learning ideas like loss functions, to basically score how biologically plausible the AI's final 3D reconstruction looks.
And this is where the biologists come back in heavily.
Exactly. The experts help define what a reasonable or correct 3D nerve cell structure should actually look like. They provide the standards the AI can measure its own output against.
Okay, so that's the technical game plan, but how do they actually plan to build this thing? What about practical implementation?
Yeah, the sources touch on several key points there. First, you need a really clear definition of the core scientific question this AI tool is ultimately going to help answer.
Wait, what's the point?
What's the point, exactly? Then, assessing if you actually have enough data or if you can realistically get it, that's crucial. You also have to match the technical challenge to your team's actual capabilities. Can they build this?
And justifying the effort, I suppose, the potential scientific impact.
Too, yeah. That's part of the justification for the project.
Data needs seem like a really big one here. They start with those 7 labeled cells.
Right, but they know it's not enough. They suggest looking into things like semi-supervised learning techniques.
Semi-supervised. That means the AI can learn from unlabeled data, too.
Potentially, yes, which could significantly cut down on the incredibly tedious manual labeling work needed. It's a smart approach.
So they need more training examples somehow.
They suggest maybe data augmentation, creating slightly altered versions of the images they do have, or maybe comparing their results against commercial models to generate more training material. Get creative.
And the system design itself, how should it be structured?
They recommend a modular approach. Keep the segmentation part, the tracking part, and the final reconstruction part sort of separate.
Easier to build and fix things that way.
Yeah, makes it easier to develop and troubleshoot each piece independently. And, importantly, build in an interface right from the start. An interface that lets those biological experts easily jump in, check the quality, review the results, and make manual corrections if they need to.
Which loops us right back to those fundamental technical challenges that make this expert oversight necessary.
Absolutely. That difficulty in creating standard objective definitions for these super irregular cells, that's probably the biggest conceptual hurdle for the AI.
It's not like finding a simple circle, like a cell nucleus.
Exactly. Defining these complex, tangled shapes precisely enough for an algorithm is really tough. It just highlights why that close collaboration with biologists to try and establish consistent standards is completely non-negotiable.
And validating the final 3D models sounds equally tricky.
It is. With limited, perfectly labeled ground truth data, there isn't always one single objective correct answer for every reconstruction. So the validation criteria often have to be based on biological plausibility. Does the output look like a functional nerve structure according to an expert biologist?
So overall, it's just a lot more complex than standard image recognition tasks.
Significantly higher technical complexity, yeah. Because of those intricate shapes, the way the cells are all intertwined, it just demands much more sophisticated algorithms than simple pattern finding.
Okay, so pulling all this together from the sources,
Yeah.
what's the final verdict? What's the main conclusion?
The clear takeaway, despite all those challenges we've discussed, is that they assess this research as technically feasible.
It's hard, but possible.
Hard, but possible. They believe it's solvable with the right approach.
And that recommended approach is step by step.
Exactly. A phased, incremental approach. Start small, maybe build a proof of concept for just one part, and then gradually build up the complexity.
And the main development focus, initially at least,
Should be on building a really robust foundation for that 2D image segmentation. Getting those initial boundaries right in each slice is absolutely critical. You could argue that's maybe the bigger technical hurdle than the step of stacking them up into 3D afterwards.
And they suggest looking at existing work.
They specifically suggest looking at and referencing existing open source image recognition models as a potential starting point. Don't reinvent the wheel entirely. And crucially, heavily integrate both that expert biological knowledge and the precious training data they have or can generate to make sure the results are actually biologically accurate.
Okay, now let's connect this to the image that came with the sources, because seeing it really helps, I think.
Yeah, it gives you a powerful visual insight into what they're grappling with and what the end goal looks like.
What are we seeing in the different parts?
Well, you can see different views. In the top left and bottom left sections, those likely show examples of the segmented structures within the 2D slices. See the different colors. Those might represent different identified cells or parts of structures that the AI has found.
So that's the AI's work on the flat images, identifying those boundaries.
Exactly. And then in the bottom right,
That's the 3D representation.
that's the payoff. That's the 3D model they're ultimately trying to build.
I see.
So the image really demonstrates the whole process, starting with those flat, segmented 2D inputs and moving towards the final volumetric reconstruction that the AI system is designed to produce. Makes the abstract goal very concrete.
And there you have it, a deep dive into applying AI to reconstruct these incredibly complex biological structures from electron microscope images, all based on the source material provided.
Yeah, we've walked through the intense challenge, the nature of the data itself, why it's such a puzzle for AI, and that proposed technical roadmap for how they might actually solve it.
Hopefully that gives you, our listener, a concise but pretty thorough understanding of this specific, really cutting-edge problem in neuroscience research.
And it does leave us with a fascinating question, doesn't it? Given the extreme complexity of these biological structures and the clear need to integrate expert biological knowledge just to validate the results, how much will AI like this truly replace human experts in scientific analysis? Or is it more about augmenting their abilities, becoming an essential, incredibly powerful tool?
That's a great question. Or, thinking bigger, if they are successful, if they build an open source tool that can reconstruct structures this intricate, how might that kind of innovation speed up discovery? Not just in neuroscience, but maybe in other scientific fields facing similar visual data challenges. Something to think about.